Uniprot versus the PDB. Both of these have really important information about proteins, but there are some key differences. Each entry in Uniprot corresponds to a protein, whereas each entry in the PDB corresponds to a structure. Technically speaking, structural model. And so, like an X-ray crystallography structure or a cryo-EM structure, or they actually have AlphaFold predicted structures these days. These structures could be protein, they could be RNA, they could be DNA, and they could be protein bound to RNA or protein bound to DNA, or proteins bound to another protein, or proteins bound to a drug. All of those are things that you might find in the PDB, but in Uniprot, all you're going to find is entries for individual proteins. And as we'll get into a minute, it's actually a protein chain rather than a protein. So a protein might be made up of multiple chains. And for now, we're just going to use this term protein. So Uniprot entry corresponds to a protein. And in the case of a monomeric protein where it only has a single chain, then that's true. But if you have something that is heteromeric, then it's going to have multiple different Uniprot entries associated with it. But for now, let's just stick with the word protein. We'll get into the complications in a minute. Take, for example, AGO2. So AGO2 is Argonaut 2, and it's a protein that I work with in grad school. It's involved in RNA interference, and what it does is it binds to these small RNAs, microRNAs, or siRNAs, and it goes and it finds the messenger RNAs that have a complementary sequence, and it binds them, and then it alters the translation of those messenger RNAs. And typically, it's kind of shut down the production of proteins there. And so this is the structure of an Argonaut, and so you can see that we have this protein, and it's bound to this RNA. This was actually solved by Ilato Kayem in my grad school lab, Limor Joshua Tor's lab. And so I'm very familiar with this structure. And this is its PDB identification number. Note that they're going to these longer numbers because they're running out of the shorter ones. If I search for AGO2 in Uniprot, what I see is that I get all of these entries. This is the Uniprot identifier, and you can see that I have AGO2s for all the different species. This is the entry for the human Argonaut 2 protein. You can see that's going to tell me about its function. It's going to tell me about modifications and key sites. The great thing about Uniprot is it links out to a lot of information as well. One of the places that it links out to is actually the PDB. And so if you're in a Uniprot entry and you scroll down, you'll get to this section that's structure. You can also get to it by clicking over here. What you'll notice is that the first thing that we see does not look anything like what we are looking at over here in the PDB. This is because we're just looking at a single domain. So it's pulling in the PDB entries that are associated with this protein. And right now, what we're looking at here, because they're in alphabetical order based on the identifier, this is actually only a portion of this protein. So this is only just a little bit of this protein, a single domain of this protein. The one that we are looking at over there was this one, which had the full length, or mostly full length sequence of the protein. We could actually, if we were to click here, go and look at it in the PDB. And note that there are different options for the PDB, depending on which one you want to go to, whether the European one, the RCSB PDB is the one I typically use. That's the one that's hosted by Rutgers in the United States. So basically, you can see that there's multiple structures that are associated with this single protein. It'll tell you some information about it, such as the resolution and the sequence that it contains, but it can't tell you here if it contains anything else, if there are modifications, mutations, things like this. So you'd actually have to go to the PDB in order to figure that out. But if you come over to the PDB and I were to search the PDB for AGO2, this is what I find. And so you can see that some of it's not even I could have done a better search to make sure that I got only what I wanted. But even when I go and I find what I wanted, now you can see that there's various Argonaut structures that correspond to Argonaut domains, individual domains, things that are binding to other ligands, all these various things. And so each of these structures that's been solved has a separate PDB entry. And in that PDB entry, I can go and I can play around with the structure in 3D and just kind of this simple viewer, so this Molstar viewer, but I could also download the files and open them up in something like Pymol. And you can also just fetch it directly from in Pymol using this code. It'll tell you information about the experiment and about various things like that. But this is all associated with this structure. And so if I actually come over here to the sequence, you see there's these different chains. And so right now I'm looking at the protein, but I can also look at the RNA, find information about the RNA. And so each of these kind of will have multiple chains. And so it's a little confusing because the PDB uses chain to refer to any sort of separate thing, separate object inside of the structure, but not all of those chains are actually protein chains. So when I'm talking about a protein chain in the context of a protein, that's going to be, it has its own gene and it's translated as its separate thing. Even though the functional protein might be made up of multiple chains, each of those chains would have its own entry in Uniprot, as long as each of those chains had its own gene. If you have a protein that is homomeric or something like this, where it has chains that are the same, but multiple copies of them, you'd have a single entry that corresponds to one copy of it, basically. In a Uniprot entry, it's just kind of a single protein chain. This is most evident if you have a multi-chain protein. So not only would we not find a entry for a protein bound to RNA in Uniprot, we would also not find an entry for a multimeric protein, or at least not the full thing. And so if we think about, let's do hemoglobin. So hemoglobin is made up of 4 subunits, and 2 of them are the same. So you have the alpha subunits and the beta subunits. And then there's also delta and epsilon and gamma and things. But the main ones in adult hemoglobin are going to be your alpha and your beta. And each of these chains is thus going to have its own entry. And so you can see that there's all these various minor forms of hemoglobin chains, but then you've got your alpha and you've got your beta. And each of these is going to have a separate entry in Uniprot because they're different proteins. They're different protein chains. They're translated separately. And so each thing that's translated separately is going to have its own Uniprot entry. But if I come in and I'm looking at the structure, well, now I'm actually seeing the whole thing. And so it's actually showing me both the alpha and the beta chain in this structure. And if I were to come over here and I were to search for hemoglobin, it's going to show me things that are actually this hologlobin, so things that actually have multiple chains. This, however, so it'll show you the structures of things that don't correspond solely to this entry. But this entry, the rest of the entry, is going to correspond solely to that chain that we're thinking about here, this beta chain. So all of the information that's given about the sequence, about all of this various things, is going to be for that beta chain. But then it pulls in structures from the PDB that contain that protein, that contain that chain, which is why you're seeing that you actually have these other chains and other things that are in the structures that you're seeing in Uniprot, even though the entry itself is only showing you, say, the sequence for that chain. Uniprot is really great for a lot of different things if you want to know about that chain, if you want to know about that protein. And the PDB is really great if you want to know about the structure. And so again, each entry in the PDB is going to correspond to a structure. And so each of these would be a structure of hemoglobin. And each of these would be a structure of Argonaut. And some of them are bound to things and some of them are single domains, so only parts of the protein. You can have proteins with multiple chains. You can have proteins bound to things. And although it's called the PDB, the protein data bank, there's also structures of things that are not just proteins. Most of the structures, though, are proteins, at least I think so. And you can actually just go, the PDB is a really great website. And if you go, you can just kind of go and look and see what's new. They have a lot of educational resources as well. So lots of things in the PDB that are really great, but it's important to keep in mind that those are going to be entries for entire like structures, and those structures might contain multiple things. They might correspond to multiple Uniprot entries. So as we see, if we search our PDB for hemoglobin, a single PDB structure can correspond to multiple Uniprot entries. And so if we go and we look at sequence here, we can see that we have these two chains of the alpha and two chains of the beta. And so each of those is going to have its own Uniprot entry associated with it. And if I go to the structure summary page and I scroll down, it actually is going to give me this link so I can go and I can find these chains on Uniprot. So I have the Uniprot identifier for both the alpha chain as well as for the beta chain. And note here that I'm saying chain, but it's also, there's multiple chains because there's two copies of it in this structure. And so that's what this BD and this AC refers to. And if I'm in the structure viewer, I can go and I can choose what kind of chain sequence, which chain sequence I want to display on here, and then which copy of that chain, if there were differences. That was a fairly simple example where we only had two different proteins involved. This one is going to get a lot more complex. So this is another structure that I am very familiar with. This structure is the structure of the human mitochondrial ribosome, its large subunit. And so our mitochondria have their own ribosomes, which is really cool. But what's not so cool is that those ribosomes, because they come from bacterial ribosomes, they are also subject to inhibition by some antibiotics that actually target bacterial ribosomes. And so an example of this is linezolid and other oxazolidinones. And so you can see that this is the structure that of the linezolid bound to this mitochondrial ribosome. So this is actually a structure from my postdoc work with Denitsa Fujimori at UCSF. And so we're collaborating with Jamie Fraser in his lab, and Tushar Raskar was the one who actually solved the structure using cryo-EM based on the mitochondria that I purified for him. And so it's purified in the context where it's containing this antibiotic, so we could better see how this antibiotic binds to this ribosome. And hopefully this complex will help people better design antibiotics that don't inhibit our mitochondrial ribosomes, but still inhibit the bacterial ones. But you can see we've got a lot of things going on. And so the ribosome is actually made up of a lot of different proteins, as well as different RNAs. And then you also have some like tRNAs and various things like that, zinc, magnesium, water, all of these things are going to be kind of found in this structure. And so you're going to have a lot of different Uniprot entries that would be associated with these. And so if I were to actually go and I were to click on one of these chains, as long as you click on one of the protein chains. So you're not going to find entries for RNA or DNA or anything like that in Uniprot, but you will find entries for each of the individual protein chains. You can see it's actually going to show me the Uniprot entry that's associated with that. And so it has a Uniprot entry that's associated with that individual chain rather than a Uniprot entry that's associated with the entire mitoribosome. So our PDB page was for a structure, and so it cannot be associated with multiple Uniprot entries if you have a structure that contains multiple protein chains. Each protein chain has its own Uniprot entry. So Uniprot entries correspond to protein chains. So each of the hemoglobin chains has its own entry, and then you'll have entries for the hemoglobins that chain in all sorts of different species. In the PDB, It has structures, and those structures are often of proteins, but not always. And those structures might contain multiple proteins. They might contain protein and DNA, protein and RNA, RNA and DNA alone. They could have lipids, all sorts of various things, despite the name, the Protein Data Bank. At the end of the day, both of them are going to have really important information. Uniprot, I love. It's kind of like your first stop for proteins information, and you can do all sorts of different things. And so on an entry, if you go down to like sequence, you can go to tools and you can do all sorts of things with it. You can compare it to other sequences. You can get information about it, like its isoelectric point and things like this. You can compare sequences. Use these tools. You can blast, align, search. You can save things to your basket. So just a little plug for Uniprot is a free resource. These are both free resources that are really, really helpful for when you want to find information about a protein or about a structure in the case of the PDB. All sorts of really cool things that you could do with Uniprot. And so I highly, highly recommend, and I've got more resources about how to use it and the PDB.